information in it. And the information that they told you about that professor, he lived in Kenya and he studied sickle cell. And the information he told you from the movie was that originally, and when we studied sickle cell in class back in October, then again in November, and then again one more time in January, when we looked at it, all we looked at was sickle cell trait and sickle cell disease, right? And we said we couldn't tell the difference between having a trait and being homozygous dominant. You can't tell the difference because they're both don't show symptoms, right? Okay. Well, this professor came up with a chemical test that shows you the difference between sickle cell, those with sickle cell trait and those that are homozygous dominant. He added a chemical A, let's call it, that into the blood sample that took away the oxygen. And since the oxygen was gone, if it had a trait, then the cells would sickle. They'd change of shape. So that told you the difference. That showed you a phenotype for the heterozygous. And that showed you and of course, the dominant homozygous had a different phenotype. So the first phenotype, little s, shows up when there's just normal red blood sample. Blood sample shows you sickle cell. You know it's little s. If your normal sample, when you add chemical A, sickles, then you know it's big S, little s. And if it doesn't sickle, you know it's big S, big S. Are we clear? So you have three phenotypes now. Now, you remember this 9:3:3:1 ratio from our genetics work, and you know that ratio is for a heterozygous cross, and this is a phenotype. So this is a phenotypic ratio. And the reason this isn't so big, this one's so big, is because this includes both hetero, thank you, and homo. You can use it if you want. Anybody that doesn't have a physical copy of the lab can go to your Jupiter grades and open up the copy on your screen.
Can I get another laptop?
There's a plug right here.
Just the lab, right?
That's the lab. Also, you can share with the person next to you. You can share with the person next to you, whatever you want to do. All right, so here you have hetero and homo. That's 9. That's why this is so big. But now we have 3 different phenotypes. This 9 isn't going to be there. It's going to be some other ratio. So we actually have to do the dihybrid cross. You're going to have to do that on Monday. When you do the dihybrid cross, this is the way it looks. So most people see this and they understand how to read this stuff here. But where most of you get it wrong is how you figure out these 4 gametes. And the other thing that most people forget is the IA, IO, IB genotype, ABO blood type. And it's frustrating because Mr. Desipeta covered it, I covered it, you did it on Study Island. This should not be new, but for some reason, and I think it's lack of effort for the most part, people are still kind of like, I don't know what's going on. So IA, there's 3 possible alleles, IB and IO. 3 possible alleles. These are all one gene, gene 1. Do you understand that? That's blood type. Sickle cell has 2 alleles. It's either a big S or a little s. That's it. 2 alleles for gene 2. Do you see that? There's 2 genes. One of them is the IA, the other is the S, big S or little s. All right. If you read the problem, they tell you the whole population's A blood type. So that means that there's no IB, right?
Right.
If you read the problem, let's read it together. It says in number 4, I'm still working on number 4 here, and we're going to keep switching before 4 and question 4 and 12 because they're related to one another. In humans, blood type is a result of multiple alleles, as we just said. 2 parents that are heterozygous for type A. How do you get heterozygous for type A? What's the only way to be heterozygous? What is type A? Is type A a phenotype or a genotype? It's phenotype. It's a phenotype. That's it. When they say Heterozygous, that tells you what? That tells you genotype, which tells you what? You have what? IAIO is correct. How does he know it's IAIO and not IAIA? Because it's hetero. Good. How do you know that it's not IAIB? IAIB is hetero.
Because they know bees.
Because what phenotype would you have if you had IAIB? AB. You had an AB blood type. You're right. It's codominant. It would be AB blood type. All right. So that's how they know. I see a lot of people are quiet. I see you. Remember, you need to be able to answer these questions and figure this out. That's how we just figured this out. That is how you figure it out. All right, so that is IAIO. That's the phenotype for A blood type. Now that's one gene. That's gene 1. What about gene 2? What about sickle? It says what? What does it say?
Number 4, right?
Number 4. It says what? The sickle cell trait. They have sickle cell trait.
So they hetero.
That means they're heterozygous. So what is it?
Big S, little
The
S.
big S, little s. That's right. They are carriers, and the reason they're carriers is because it's a recessive trait. If they were heterozygous and it was a dominant trait, then you couldn't have a carrier. If you had the big one, you got the disease. All right, so that's it. We know what. So what is the phenotype of the parent? What is the answer to 4A? What's the phenotype of the parent? Or sorry, the genotype. Thank you. What's the genotype of the parent? IA. IO, big S, little s. The parent, very good, by the way, excellent answer. So you have 2 genes, and you have 2 copies of each gene, right? And they're both heterozygous. How did we get these gametes? Notice you only have 1 copy of each gene in the gametes, but in the offspring, the probable offspring, the possible offsprings, you have 2 copies of each gene. These are diploid organisms. These are full-fledged organisms. These are your sex cells. Are we clear? So every time you see this, that's what you should see. You should see it in your mind. If you don't, you need to come and get clarification. All right, so now how do we get these? We use the FOIL method. Miss Simpson covered it with you, or Mr. Hogan, or whoever. Somebody in your history covered the FOIL method, your academic history. So how do we get what is FOIL? What am I going to do? First, so basically IA goes with, the first over here, goes with big S, right? IA, big S. So first outside. IA, little s. And then IA, what is it? Little s. So that's where I get this one. You see IA, little s. You see that? And now the next one is inside. I'm going to use a different color. IO big S. I like to use the arrows on the bottom. It's important that you make sure you're clear that you're getting these right. This is where most of you mess this up. If you make a mistake, this is where you make your mistake. It's either in getting the parent's genotype, you make a mistake, or when you're coming to the gametes. This part is easy for everybody. So the next one's IO what? Little s. And so here you have them. IAS, IA, little s, IO, big S, IO, little s. There's your gametes. And you notice it's the same gametes on the left because both parents are heterozygous for both genes. And that would be the answer to B, right, for B. This is obviously the answer for the dihybrid cross. So now we're flipping to page 12. Sorry, not page 12, but number 12, question number 12, which is on page 10. On page 10, let me pause this for a second. So here you have, we've done the FOIL method, we've come up with both of the gametes, we filled in this chart. If you don't have it, make sure it's filled in for you so you can reference it for home. And then we have this. On your chart on question 12, it says probabilities. Well, it doesn't say that. It says blood test results, right? And you have six of these, right? One, 2, 3, 4, and then 5, and then 6, right? You see that? And in here you have your results for each one, and I'm not going to write them all in there. It says type A, normal red blood cells, type O, normal red blood cells, type A, sickle cell trait, type O, sickle cell trait. Now there's numbers here. The numbers are 48. I'm going to write these in because they're easy to write. 18, 92, 33, 27, and 6. So those are my 6, right? What are these? What is the number of children? What is that?
Who have it? Who have those results?
Yeah. So what would you call that for when we're talking about chi-square? These have you observed. And when you're talking about blood tests, results, what we call that?
Expected.
Look at your chart. Those are not expected, no. Phenotype, that's right. So this would be your phenotype. And then what would you call the probability of that phenotype?
Expected.
It's almost expected, almost expected. Not quite the expected yet. What number are we missing to help us find the expected? Somebody said it, the total. You're right, so you got to have confidence. The total, you have to have your total, because what is the probability going to give you? It's going to give you a percentage, that's right. And a percentage doesn't help you. You need a number. This is not a percentage. Is this a percentage? No, it's a number of students. So you need to know, number of children. And if you're going to find out, you need to find out the expected, you need to find out the number of expected. So in fact, if you want, you could add another column. That's your probability. Here's your probability right here. You're going to write those in. And then you could add another column that could be what? You're actually expected, which is going to be your probability times times your 224. Right, your percentage times 224. We understand that so far? Where do we get our probabilities? We get them from our Punnett squares. That's it. So let's count them. Let's do it. Let's do the first one right here. The first one says what? Type A normal red blood cells. Which one of these would be normal? Yeah, Curry. Normal red blood cells. The type A is either going to be what? IA or IAIA or IAIO, right? There's only two blood types here. It's either A or O. The A's have two ways of getting it. What about normal versus sickle cell? What do we got here? If I tell you it's normal blood type, what is the genotype you should be looking for to count? Big S, big S. Why not big S, little s?
Because that's a carrier.
That's a carrier, that's a heterozygous. Now, let me be clear. On a standardized exam, they may not give you this blood type. They might tell you percentages. They might have you include the heterozygous with the homozygous because you might not be able to tell the difference. Do you see what I'm saying? So you got to be careful. You got to read the question carefully. In this question, they tell you you can tell the difference between the heterozygous and the homozygous dominant. Are we clear? So we can separate them. If you can't separate them, then it's 9-3 to 3-1. If you can't separate them, then you can't use that option. Does that make sense? Because we're talking about phenotype. All right. So let's go ahead and do these. The first one says type A normal red blood cells. Where are they? Type A normal red blood cells. Where are they in this mess?
Yes, the green ones.
So IAIO or IAIA and big S, big S. So this one, This 1,
2,
2,
3,
3. So how many do I have?
3.
Out of what?
224?
3 out of 16.
16.
Which gets you what percentage? What decimal? Somebody do the math.
I don't know. Sorry. I don't remember.
Is it 18 or 19?
If you round it, it's
Round it.
then it's
19. Let's go with 19, just for now. So that's 19%. 19% of what? 19% of what? No. Of 224. So what we see, you guys you're not making connections, and I don't know what I can do to help you make connections. That's why we did that dragon package, and I hit it so hard. That's why we did the Dihybrid Cross so hard, because I knew this was coming. You got to make connections, all right? You got, these are what? What do these represent? The number of children out of 16, so the percentage of any population. So the population is 224, so 3 out of 16 is 19% of 224. Because that's the population. So it's 0.19 times what? 224,
equal 42.
which is 42. All right, so when we're looking for number 1, and so 42 is that's our expected. That's the expected. You can write that down. That would be the expected for the first row. So when I look at my row, when I look at the big chart, yeah, I told you, look at the phenotype. Look at the second column. That's what you have to do for problem 12, is fill it in. What's the phenotype there? What goes into phenotype? No, type A, normal red blood cells, type O, normal red blood cells, etc., right? Fill it in if you don't have it filled in. Now's the time to do it.
Can we put the gene?
No, put the phenotype only, please. The observed is what? The number. It's right, just copy and paste, people. Get it written now. The expected is what? In the first row, it's 42. It's this number that we're calculating. It's not the frequency. The frequency is what we have to use to get the number. So, all right, so that takes care of our first row. The second row asks for the type O normal red blood cell. Where's type O? How do you get type O? I-O. How do you get normal?
Big S.
So you have to have I-O, big S. How many in here have I-O, big S?
The blue one.
The one blue one. So that's 1 out of 16, which is what? Do the calculation, somebody. 0.0625. When you see that, you should automatically see 6.25%. At this point in your life, you should be able to see 6.25% there. You shouldn't have to wait for someone to tell you what that is. Okay?
Yeah.
So you should see 6.25%. So 6.25% of what? 224. So 0.0625 times 224.
Equal 14.
So that's 14. So your first one was, what was the first one?
48.
48. Your second one's what? 14. All right.
No.
Why did someone say 48?
I was looking at observed.
43. 42. Get it straight on the quiz though. And then what's the next one you said? 14. So now the third one says type A sickle cell trait. Type A could be IAIA or IAIO, but sickle cell trait means what?
Big S, little S.
Big S, little s. Little s gets you what?
The affected.
The affected, the disease. It's okay, you got it right. It's good to get it wrong now. This is good. You don't want to do it on the test, all right? So when they say you got the disease, that means you have both little s's. And by the way, I think you're doing really good coming back from that little trouble you had. Okay, I want to mention that. All right, so big S. So you try to do. I say. Well, again, we're on the third one. It says big A sickle cell trait. Which one? What? Where are they? What color do we have here?
Pink.
So we're looking at big S, little s with a big A of some kind. Big S, little s with a big A. So we're looking at this pink, right?
Yeah.
So 1, 2, 3, 4, 5, 6. 6 out of 12.
Huh? 16.
6 out of 16. Sorry, my bad. Somebody do the math.
Justin. 0.375.
0.37. 0.38. Okay. 38% of what? Notice how I already knew it was 38%. I didn't have to I didn't have to change it. Of 224. What's that number?
84.
So it's 84 is what I expect there. So now the fourth one says type O sickle cell trait. How do I get type O? I-O, and then sickle cell trait. What am I? So I-O, big S, little s. We get how many?
2.
2. So 2 out of 16 is what? What is it?
0.13.
0.13. So then we're dealing with what? 13%? So what's 13% of what? 224, the total.
28.
So
Well,
it,
when you
when you multiply, what do you get?
28.
28. Okay. So are we getting this?
Yeah.
All right, so the next one says, what's the next? Someone help me out here. Type A sickle cell disease.
So little s, little s.
Little s with what?
Type, IA.
Right, either one. So here's 1, here's 2, here's 3. So we're talking about the purple, right?
Yeah.
So what's the 3 out of what?
16.
3 out of 16. So what is it? 19%. And 19% of what? 224. So we got 41 expected there. So what's the last one? Type O sickle cell disease. Little s for sure. And then type O, how many do we have? 1 out of 16. And so we get what?
65 again.
We get 0.62065, so 6.25%. I see your hands up. Just give me a second. 0.0625 times 224 is 14. And those are expected values. Question and up was up first and then yours. Yeah.
The 41 and 42, the first one and the second to last one, it's the same numbers, but you get different outcomes.
You see that? Why? This is what we're talking about. Why is this one different from this one? Why is this one say 41, this one 42? Why? Somebody tell me the answer.
Where you at? Huh?
There's a simple answer. It's a problem that happens a lot in science that we always have to try to address. That's why we deal with significant digits. That's why we try to hit you about how to do this one thing that they taught you in math. What's the thing that we're doing wrong here? What error are we dealing with? Somebody said it. Rounding.
Somebody said
It's a rounding error. Somebody multiply 0.19 times 224. What exact number do you get? 42.56. What happened? Somebody take 3 and divide it by 16. .1875, where most people round it off to 19, right? Somebody probably multiplied times 0.18, and what they ended up getting is something closer to 41. And what's our number for 19? 19 times 224, what was it? 42 something or 41 something?
42.5.
So we'll just go with 42, although it sounds like it should be 43. All right? Do you agree it should be 43? Do you just want to correct it now? Let's just correct it now and just keep it 43. So when we do this, this is the problem with error, with rounding error. That's why we have to try to make sure we correct that before we get our numbers mixed up. Everybody okay with that?
Yeah.
All right, so we're at 43. That's our expected for each one of those. Because of rounding error, these may add up to 224. They might add up to a little bit more, a little bit less, right? But we're good with the idea that as long as we're close, we're good. Yeah, question. Then you. There's a question. Okay, yeah? I can't hear you. When you did 0.1875, so that, see, there's your difference. That's it. One would be, if you didn't round it, it'd be 42. If you rounded, it'd be 43. So which number do you think we'd probably want to use?
42.
Let's use the 42. You know what? It's just simple science. You have to work this out.
If you use 43, it would be 226.
All right. So we're good. So we're close enough. We get the idea. This is what you're gonna have to do. Hopefully, you're not making rounding errors. Here's my suggestion: always use the unrounded number in your calculator when you're doing your calculations. And then, if you want to put 0.19, I wouldn't even write 0.19 here. I would go ahead and put 0.1875. I just didn't have room to put it all. All right, we're good. We're not done yet, though, are we? We're only halfway through the problem. We're only halfway through the problem. Now, granted, I'm explaining everything, and you would probably do it a lot faster if you were on your own doing it on a piece of paper, assuming that you know what you're doing. If you don't know what you're doing, then it's even faster because you have a blank piece of paper. Anyways, sorry for the joke, but I don't know what else to do. Sometimes it's either you cry or you laugh, and I guess I'm at the laughing stage. All right, so if we go ahead and we take a look at what we got, you guys have it written down. We got our expected, we have our observed, we have our phenotype. How do we find chi-square, whose symbol looks like an X, but we know is not? Let's take the sum of what? It's the sum of the observed minus the expected squared divided by the expected for our first row, right? For our first phenotype, plus what? The same thing, observed, not the same numbers though, but the same idea. Observed minus expected squared divided by expected for the second possibility, right? For our second phenotype. And then we're going to do it again. Well, you guys should be writing this down, and you should be doing it. For our third phenotype. And we're going to do it again for our fourth phenotype. It gets long, doesn't it?
Don't you just want to put etc.?
You could, but people have been complaining that they don't understand what's going on. So I've been kind of trying to, let me just say this, 1, 2, just so you don't think that's part of the formula. I'm just keeping count there. And then I have to go ahead and do observed minus expected for which? For the fifth.
Oh.
For the fifth one, right?
Yeah.
For the fifth sample.
N6.
And then finally, it's going to be the observed minus expected squared divided by the expected for our final sixth. All right, so that's your chi-square. If you do all that and you do the math right, you get a chi-square. Could someone please calculate that now, please?
All of that.
All that. Let's get it done. Let's fill it in. You want to fill in and let's fill in together. So it's going to be, the first one's going to be, I need the numbers, so I don't have the numbers.
Minus 42.
So it's 48
6.
minus 42 divided by what? 42, right? Oh, wait, no, we messed that up. We have to square this, right? Square 48 minus 42, then divide that answer by 42.
So 6 squared is
0.86, you say. And you guys do it anyways, even though you see the number up here, do it on a calculator, see if you get the same answer.
Okay.
We saw some mistakes already. So the second one is what? Observed is what? For the second one should be 18, observed is 18, minus 14, square it, divided by 14. The next one's what? The next one's 92 minus, what was it? Minus 84, square that, and divide it by, 84. The next one is 33 minus what? 28, square that, divided by 28. And then the next one's 27 minus what? Minus 43. Sorry, 42. Somebody didn't fix their answer. That's right. 42 squared. Shouldn't there have been another 42? Didn't we go through that? Oh, I see it. Yeah, thank you. Divided by 42. And then the last one, 6 minus what? 14. Square that, divided by 14. Now, interesting. Do you see why we square this number? Because look, you start to do this, what are you going to get? A negative. You square it, turns positive. So it just keeps everything positive. So we're summing everything up. That's why they square this number. All right. We do that a lot in math and science. All right, so our first decimal is 0.86. What do we do with our second decimal? Point what? 57. What's the second number? Third number, sorry. By the way, this is like, goes there, goes there, goes there. What's our third one? I should hear little clickety clicks of calculators all throughout the room. Come on. There's calculators in there. There's computers. There's cell phones. Come on. I should get at least 5 people doing the math and 5 people agreeing. The third digit, the third one, what is it?
The second one is wrong. It's 1.1.
Oh.
What's the second one?
The second one is what?
0.14.
0.14.
No.
What is it?
1.14.
1.14. Jehoshaphat. All right, what's the next one? 0.76. The fourth one? What is it?
It's like 0.9.
Point. So you're saying 1? All right, so you're rounding it.
No, it's 0.9, but I rounded it like.
All right, so 0.9. Okay, go ahead. Next. The fifth one? 27 minus 42 squared divided by 42. What is it? 5.36. And the last one? 4.57. All right, add these together. What do you get? Our chi-square equals what? Equals what? You guys got to get faster on these calculators. What is it? You added these up, you got 21? I don't see that being up to 21.
It's 13.51.
Yes, 13.59.
13. 0.59. You know what? This is a good opportunity when you get numbers like that. Look, I've got numbers like that too. I've made mistakes. 13.59. When you get numbers like that, and you're like 21. You have to look at this. You have to look at it and think, does it make sense that this equals 20 something? No, it don't. So I made a mistake. Let me go back and see where I made it. All right. So 13.59 is your chi-square. Are you done? No, you're not. All right, so now okay, so see, that was all that. Now let's, degrees of freedom is correct, so now, it's okay, let's try to focus, though. We gotta figure, this is only part of the problem. This is only part of the problem. What is it we have to do now that we got chi-square? What do we have to do? We have to think about our graph. We have to think about our tail, and we have to think about our rejection area, and we have to try to find our critical value. Hold on, let's talk about it. Let's think about it. Our critical value. Let's think about our rejection area. And it's just hard for me to believe you're asking for a rescue pass at this point. Because we're like in the middle of a very important piece of this. Fill it out completely, please. All right. The time is 3:08 by the time you're done filling it out. All right, so we need to find our critical value. We need to find what the rejection area is. And in order to find the critical value, we need two more numbers. You're going to have to find your p-value, and you're going to have to find your, what is the other one that somebody said?
Freedom.
Degrees of freedom.
Degrees of freedom.
Right, degrees
I couldn't think of the D word.
of freedom. Bless you. So the degrees of freedom. Now, the degrees of freedom at this point might be easy for you. How many different possibilities did we have? 6. You see the 6 here? There's 6 of them. Not the 224. 224 is a total, right? That's a total individuals. But how many different possibilities were there? There were 6, right? 6 different phenotypes. So it's 6. Remember, degrees of freedom is n minus 1. So what is it? 6 minus 1, which is 5. So 5 is your degrees of freedom. That's it.
What N represent again?
N is just the number of possibilities.
Oh, okay.
So when we're dealing with P, though, what's P? Unless you're told differently, you're going to assume that P is 0.05. 0.05. You're going to assume 0.05. Remember, P can also be declared as alpha, okay?
Oh.
A or alpha is 0.05. So now all you got to do is go to your chart. Go to your chart on page 12. Find degrees of freedom of 5. Find your p-value of 0.05. What number do you have?
11.070.
That's your critical value. That's it.
What? That's 5 degrees of freedom. Oh wait, I'm tripping. I'm sorry. N minus 1. My bad.
All right. So we got critical value is 11. I'm glad you make. Hey, look, it's not your bad. I'm happy you're saying it out loud, so you hear yourself. Other people hear you, and they're like, okay, that's wrong. That's not how you do it. So this is how we learn. This is a process. All right, so it's 11.070, 13.59 is our chi-square. Is our chi-square bigger than our critical value? Before you answer, let me remind you that this is your critical value here. Do you see it? It's kind of hard to see there, so let me put a little blue line. This is your critical value. This is small, right? It gets bigger this way. All right. If your critical value, if your chi-square, which is here, 13.59, is bigger than your critical value, which is equal to 11, it's going to be somewhere in here, right?
Yeah.
Which means what? You're rejecting the null hypothesis.
Prove it.
You're saying the null hypothesis is rejected. And what you're saying is, I'm going to accept the alternate hypothesis. The alternative hypothesis. When you're dealing with the null hypothesis, that's the idea that this is all the difference between what you're expected and you're observed is just due to randomness. When you see that the critical value, that the chi-square is bigger than your critical value, then you say it's not due to randomness. So the difference in alleles in this population, right? The difference in alleles in this population is due to selection. Something selecting one group over another. What does that mean? Something's killing some of these kids more often than others. That's what natural selection is, okay? That's what something's killing the babies more than some other of the babies. So knowing what you know about sickle cell and knowing what you know about this particular population and looking at this, what do you think the selection pressure is? Look at the difference between the numbers, the expected and can I go back for a second? So someone just asked, how do we know that malaria is impacting this? Look, we know malaria that gives resistance, being heterozygous for sickle cell trait, having the trait gives you an advantage. We know that based on what you did, what the video that you watched, right?
Yeah.
If you didn't watch it, you don't know. So based on the video you watched, you know that the heterozygote has an advantage with sickle cell trait. So it's because the malaria is killing the homozygous dominant. That's why you see